This video was made for the University of Georgia's Plant Breeding Practicum course for the year of 2024. So this recording is going to show me using the Gene Catch Plasmid DNA Mini Prep Kit by Epic Life Science. So, mini prepping refers to the extraction of DNA from bacterial cultures. The resulting DNA can be employed for sequencing, digestion, or PCR. So I'm going to first show you all the materials that you'll be needing for today. The first is a 2 milliliter tube. You'll also need a 1.5 milliliter tube. You'll also need some pipettes. You can see I'm using a P200 and P1000. I'm also using filter tips for both of these because I am going to be pipetting stalks. So here's what's actually in that kit. The first buffer is the MX1 resuspension buffer, which will be kept at 4 degrees Celsius because it has RNase A in it. And there's also the MX2 buffer in the kit. This is the lysis buffer, which means that it's able to rupture the cells. This MX3 buffer will neutralize the MX2 buffer, so after adding it, you'll see white clumps, which are the cell refuse. And there's also your wash buffer, which will act as your alcohol wash. We also use econospin columns, which I will link down below. Some materials outside of the kit that you'll need are a refuse for your bacterial waste, as well as low EDTA as your elution buffer, which I will link down below as to how to make. And lastly, you'll need your liquid culture, obviously. That's what we're going to be prepping today. So once you have all your materials laid out, you're going to use your liquid culture, as shown here. It should be nice and opaque, and you're going to pour that into your 2 milliliter tube. It's important that whenever you centrifuge that you make sure it's balanced. So here I only have one sample, so I'm using a balance at the opposite end of the centrifuge. You're going to want to centrifuge this for 50 seconds at 13 to 15,000 RPM. Okay, so now you can see that there's an opaque pellet at the bottom of this tube, and you're going to want to decant or pour out that liquid media like I mentioned. So here's my bacteria refuse. Okay, now comes time for the pipetting. So I use a P1000 to pipette 200 microliters of your MX1 buffer. This is your resuspension buffer, and you're essentially going to resuspend those cells that are currently at the bottom of your tube. After you've pipetted 200 microliters of your MX1 buffer into your tube, you're going to run it against a tube rack 6 to 8 times to make sure that it's well suspended. So it should be fairly frothy at this point, and that's good. So now we're going to add 250 microliters of the MX2 buffer, that lysis buffer that I mentioned. This is going to help break the cells. After adding the MX2, you're going to invert your tube 6 to 8 times and let it rest for 3 minutes. Be sure not to let it over-rest at 5 minutes or more, or else you risk over-lysing your cultures. And now it's time to neutralize that lysis buffer with your MX3. So add 350 microliters of MX3, and as soon as you do, you should see a white precipitate form. This is good. This is just the cellular refuse, all of your non-genomic cell components, and we're going to centrifuge it down again for 5 minutes. So after your 5-minute centrifuge, you're going to have a lot of cellular debris just flung against the wall of the tube. We don't want any of that, so we're going to pour the liquid alone into our econospin column. So now that it's in your econospin tube, centrifuge for 1 minute to separate the DNA bound to the column from the rest of your buffers. After discarding the flow through, that is the liquid that flows through the column, you add 750 microliters of your wash buffer. So now centrifuge that for another minute. And afterwards, you're going to transfer the column to a 1.5 milliliter tube and add your elution buffer, low EDTA. Centrifuge that for 1 minute to have your resulting plasmid prep.